So for concept check, section 1.6, the questions were: spontaneous generation was disproven by blank with his experiment using swan-necked flasks. So that would be Louis Pasteur. Polymerase chain reaction was invented in 1989-ish, so 1980s would be the decade. When a hypothesis is supported by a growing body of data that survives rigorous scrutiny, this is known as a theory. Aseptic technique was developed by, I think this is mainly talking about in surgery, that was developed by Lister. So that those are the answers to those concept questions. We'll move on to section 7. So naming, classifying, and identifying organisms. There's a lot of things listed here. Nomenclature, taxonomy, classification, so know those terms. Make a mnemonic device for remembering the taxonomic categories. Correctly write the binomial name for microorganism. Draw a diagram of the 3 domains and differentiate between traditional and molecular approaches to taxonomy. So in nomenclature, this is the naming of organisms, the scientific names that are given to organisms. And they generally use like a Latin term because there's so many languages all over the world. They picked Latin as pretty much the common name for everyone, and so they use that. So understanding and appreciation of microorganisms will be improved by learning a few general rules about how they are named. So the binomial nomenclature, it consists of 2 names. So bi means 2 and nome means name. So the 2-name naming system consists of the genus and the specific identifier. The first name comes from the genus and the second name is the specific identifier. Both names should be italicized if you write it like in a paper that's typed, Word document, in a book, in a publication. If you handwrite it, it should be underlined. We do use abbreviations sometimes, like Staph aureus is, Staphylococcus aureus is abbreviated S. aureus, or Escherichia coli is abbreviated E. coli. So the genus can be abbreviated to save space. Some of these names are really, really long, so it's like just common to say E. coli instead of Escherichia coli. So classification is the construction or constructing taxonomy. Classification attempts to make them into orderly arrangements of hierarchical groups called taxa or categories. And identification is when you discover, based on recorded traits of the organisms, we know what they are. So we are going to do some identification later in the semester. When you are given a tube of some unknown organism and you have to look at the biochemical test traits, the gram stain, the streak plate, and be able to come up with the identification of the organism. And so we have that is the process of discovering and recording the traits of the organism so they can be recognized or named and placed into a taxonomic scheme. So the levels of classification from most general to most specific are domain, kingdom, phylum or division, class, order, family, genus, and species. So you can see the most general category has several different organisms that are in that group, and they're very different. Like we have a sponge here, and a human, and a lemur, and a starfish, and a dog, and a gorilla. So those are all very different. So it's a very inclusive group, and you get more and more specific as you go down to the genus and the species. When you get the species, it's the single name that is responsible for that, or it's only one organism that fits that. So same thing here with all these different kinds of protists, and then you have some more specific categories until you get down to the very specific genus and species. So taxonomy is the science of classifying biological organisms, and it was developed by Carl von Linné in the 1700s. He laid down rules for classification but he was using like characteristics of the organism that could be seen and observed to put them into these categories. And a lot of what he did, we're finding now that some of these organisms were really not in the same category. And there's a lot of mimicry that occurs in organisms. So some things were mistakenly identified as the same species, and they're really not the same species. So genetics are, molecular genetics is used nowadays to put things into categories. But he did develop and give us an idea of how to start this whole organization of living things and classifying them into different biological species. So phylogeny is this taxonomic scheme that represents the natural relatedness between the groups. And so if you look at these, they're basically related because of their genes. And so a lot of these phylogenetic trees are now based on their genetic relatedness, how related they are according to the genes. So there used to be 5 kingdoms, and I guess most people would still recognize these 5 kingdoms, but Monera is now divided. It was back the bacteria and the archaea, but now bacteria and archaea are in 2 different domains. And Protista is no longer a kingdom. They've split that up into 6 different supergroups. So if you look back here, the bacteria and archaea both were in the same group. Now we've put them into different domains because of genetics again, because of the ribosomal RNA sequences. And we'll talk more about that in the next unit, but bacteria and archaea are really very different. And in fact, archaea are more like the eukaryotes. So the 5 kingdoms, plant, animal, protista, monera, and fungi may be the recognized kingdoms, but yeah, there's a lot of controversy about how many kingdoms we should really end up with when they get through sequencing everything and figuring out how related all the things are. So concept check 7, section 1.7, how are the names of bacteria assigned and abbreviated? What are the 8 taxonomic categories? And what are the 3 domains of the phylogenetic system of classification? So the answers, so if you paused and thought about those, now I'm going to give you what the answers are. So the names of the bacteria are assigned by their genus and their species. And so there's the genetic and the specific identifier gives us the name of the species. And the 8 taxonomic groupings, I'm going to have you list these from general to specific. And the general is, most general is domain, and then kingdom, phylum, class, order, family, genus, species. So you can come up with a mnemonic device to remember that, like dumb King Philip came over for green soup or something like that, but that would be the taxonomic groups. Help you remember domain, kingdom, phylum, class, order, family, genus, species. And the 3 domains are the bacteria, the archaea, and the eukarya. And so based on the type of cells, but again, like I said, the eukaryotes or the prokaryotes have been divided into 2 domains because of the ribosomal RNA differences. And again, we'll talk more about that in Unit 2.